In this video, I will discuss the electron transport chain. As we know, the electron transport chain is going to be where NADHs and FADH2s move through a series of proteins in order to make ATP. I really like this picture, this diagram of the electron transport chain, but you need to kind of divide it up into multiple different parts so it doesn't get too overwhelming. So the way I like to think about it is I first think about the electrons and then I think about the protons. Before we start this discussion, keep in mind that this process is occurring in the inner cristae of the mitochondria. So if you look at a picture of a mitochondria, you might notice that there's little squiggly lines in the middle. In those squiggly lines is where this is taking place. The electron transport chain consists of a series of 4 proteins in the enzyme ATP synthase at the end. What's going to happen is we are going to have NADHs enter at protein number 1, and when they enter, they will release their electrons. These electrons will travel from protein to protein until they get to protein 4. Meanwhile, our FADH2s are going to enter at the second protein. And when they do this, they're going to do the same thing. They're going to release their electrons, and these electrons will travel from protein to protein until they get to protein 4. When we look at this diagram, we can see that when NADHs and FADHs release their electrons, we're left with an NAD+ and an FAD. These molecules will go back to the Krebs cycle or back to glycolysis, so more NADHs and FADH2s can be made. Meanwhile, there's going to be protons that are going to be pumped out from the mitochondrial matrix to the inner membrane space. And we can see this on the picture by the blue little circles with the H+. These are the protons. These protons are important because they create a gradient. So as they're getting pumped out to the intermembrane space, they are getting pumped out actively, which means uses ATP. Once they're in the intermembrane space, now we have a gradient with more protons in the space than the mitochondrial matrix. This gradient is going to be what activates ATP synthase at the end, so we can make ADP and P and make ATP from it. Again, so it's the protons getting pumped out to the intermembrane space that creates a gradient that ultimately activates ATP synthase, which takes an ADP plus a P and puts them together to make ATP. The last thing we need to talk about is what happens at protein 4. At protein 4, this is where oxygen comes in. I hope you've noticed that we have not talked about oxygen at all so far in any of these processes. The electron transport chain is the only place where we directly use oxygen. So at protein 4, oxygen comes in. It's going to accept 2 electrons that have been traveling from protein to protein, and it also accepts 2 protons. When we have oxygen plus 2 electrons plus 2 protons, we're going to end up with water. So water will be one of the end products of the electron transport chain. So what do we get from this? For each NADH that enters the electron transport chain, we are going to make 3 ATPs from it. However, since we use a little bit of ATP to actively pump out protons to the intermembrane space, we will get 2.5 net ATP. When you are counting up how many ATP you get from all of these processes, you will use the net value. For FADH, it enters at the second protein, so we will get less total ATP from it. Our gross gain is going to be 2 ATPs, but again, since we use a little bit of ATP to actively pump out the protons, we'll get the net gain of 1.5.